Hi folks, well welcome to chapter 4. This is a chapter about sex chromosomes and sex determination. And I probably shouldn't say it that way because sometimes the the sex determination is not apparently a chromosome. It could be something like temperature. Uh, I suspect that it'll probably come back to a chromosome when we eventually figure out how that mechanism works. But we'll talk about some different mechanisms here. Well, first of all, I want to tell you that some organisms don't have only two sexes, and that's what we're typically think about, a male and a female sex. But some organisms have more than two sexes. And if you're when we do this lecture face to face at the traditional lecture, I'll bring a book to class. It's called Dr. Tatiana's Sex Advice to All Creation. It was a bestseller for a while. It's written by Olivia Judson, and I'll read some passages out of it, and you'll see that there are some worms out there that have arguably 11 different sexes. So I just want to let you know that the paradigm that you have right now about male and female, that's not always true with every organism, but that's where we're going to be. We're going to be talking about uh, the typical sexes, which is male and female. Now, how do you become male or female? There's different mechanisms of sex determination, and I want to show you a picture here. This is the one that you're familiar with, the top one, us, humans, and that's our XY system. By the way, it's not just mammals. Drosophila, well, fruit flies, Drosophila falls into here. They have an X and a Y, but we're going to talk about Drosophila. They do have an X and a Y, but what you're going to see is the Y really has nothing to do with the sex determination. It's really a ratio of X chromosomes to autosomes. And because if the Y is in there, that's one less X chromosome, then the ratio changes. But they still have an XY system, even though it's slightly different. You can see that a lot of other insects, like grasshoppers, they have an X nothing system. So if you're X nothing, you're a male, and if you're an XX, you're a female. So that's a system that many insects use. I realize Drosophila is an insect, and you could probably argue that Drosophila falls into this X nothing system, except for it's not nothing, it's Y. But remember, the Y really has nothing to do with it. It's really this ratio of X to autosomes. And so is there a little merging and muddying of the water between these two? Absolutely. Here's another system down here you'll see in some birds. It's called the ZW system. And what's important here is that the male's homogametic right there. So you can see that both the male has the ZZ, whereas up in mammals, the female is homogametic, XX. And in the birds, the female's heterogametic. And what am I saying here? I'm saying homogametic. That means the same type of gametes. That means that the sperm had a Z and the egg had a Z. So to be male, you are ZZ, you are homogametic. That's if you're a bird. All right. So the same type of gamete. Whereas what I'm saying here, the other word I'm saying is heterogametic. And that means that you came from two different gametes. The sperm was, the sperm had to have a Z, right? And the egg was W, and now the female is ZW. So that's the words homogametic and heterogametic. So you can see there's different systems here, and there's even more systems than just this. These are the three different chromosomal systems and birds, insects, and mammals, some major systems. We'll talk about temperature and behavior a little bit. A matter of fact, it's mentioned here. There's some systems where the temperature of the environment determines sex. That's seen in reptiles like crocodiles, alligators. The temperature of the eggs determines sex. We'll talk about clownfish and the sex is determined by the behavior of the dominant female. So, and if that dominant female dies, then a male turns into a female, and we'll talk about that a little bit. So there's behavioral interactions. But let's talk about humans for a second. On the Y chromosome, there's this SRY gene. This is the gene that makes humans male. By default, all humans start developing as females, but if they have the Y chromosome and an SRY gene, the SRY gene codes for a transcription factor. And what that means is this transcription factor turns on other genes. And it turns on other genes that basically do two things. It basically tells your Leydig cells to make testosterone, and you increase testosterone. And it tells your Sertoli cells to make this Mullerian inhibiting hormone. And what the Mullerian inhibiting hormone does is it suppresses the development of fallopian tubes and enhances or turns on the development of vas deferens. And by the way, I skipped a lot of steps here. I simplified that. So when you take my A&P class, or if you've already taken my A&P class, Anatomy and Physiology 2, you know I'm simplifying this because I went into more detail in that class. So there's many steps missed here, but that's basically what happens is the SRY gene is a transcription factor. It turns on a series of other genes that turn on some other genes because they're actually also transcription factors, where ultimately you develop Leydig cells that make testosterone, and you develop Sertoli cells that make Mullerian inhibiting hormone. And that Mullerian inhibiting hormone is extremely important to becoming a male. All right, so sex determination in Drosophila. Before I show you this, it's actually a video. I need you to remember a couple things. First of all, don't forget the central dogma. All right, you need to remember the central dogma. You need to remember that really what happens is I make this pre-messenger RNA, and this is eukaryotes. Now this is, well, it's Drosophila, so of course it's eukaryotes. Okay, so remember this is Drosophila. Remember Drosophila is a fruit fly. Remember that's a eukaryote. In the pre-messenger RNA, to become a mature messenger RNA, has to have several events occur. First of all, you have to cap it. You have to tail it, poly-A tail it. And you have to cut out the introns and splice together the exons. Introns are junk. Well, all right, so star junk, meaning what I'm going to talk about later is, maybe the introns aren't completely junk. But for purposes of for relative to the protein that's going to be translated here, that we're going to translate into, the introns are junk. None of the intron information appears in the protein. But introns might not be, quote, junk, as we'll see later in this course. So don't forget that. When we make the protein, we could have you could have two genes making proteins like this. You could have gene A. So this is gene A making protein A. You could have gene B making protein B. And then these are apoproteins, and you could end up with AA holoprotein. You could end up with AB holoprotein. You can end up with BB holoprotein. So assuming your protein has quaternary structure and is a dimer, then you could put these two polypeptides together in different ways. So you could have AA, AB, or BB. Okay, so that's something you're going to need to know for this video we're going to talk about. I need you to know that when we splice together the exons, if your pre-messenger RNA, let me give myself some space here. If my pre-messenger RNA has, let me just give it some exons. If my pre-messenger RNA looks like this, let's say it looks like L1, E1, 1, 2, 3, 4, 5, 6, 7, 8. All right? And I should have made space there. I have to do that again. I have to make space. So these things are my exons. One, two, three, 4, 5, 6, 7, 8. Dot, 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 dot. This is an intron. When I cut out my introns and I splice together my exons, I don't have to splice them together in order. I don't have to splice them together L1, E1, 1, 2, 3. I could do L1, E1, 2, 3. I could get rid of the 1, or I could do L1, E1, 3, 2, 1. Or I could do E1, L1, 2, 1, 3. So in other words, I can splice these together, these exons together in different ways. It's called alternative splicing. And what's cool is this. You could actually run into a stop codon and say, let's say exon 3 has a stop codon. Well, you're going to say, well, if exon 3 has a stop codon, if your mature messenger RNA has exon 3 in it, let's say this is what it looks like. 4, 5, 6, 7, 8. Because remember, that keeps going. Then you're really not going to make this protein. You're going to stop making the protein because there's a stop codon in exon 3. True. I completely agree with that. But if your messenger RNA got rid of that exon 3 and looks like this, now you make the protein. Because exon 3 is not there. Exon 3 has a stop codon, but that's not there. So I need you to keep alternatives like that in mind when we're learning about Drosophila sex determination. So let me see, what other basic concepts do you need to know? I got to tell you, Drosophila sex determination is pretty cool and brings together a lot of concepts. So I guess before I play the video, I'll just tell you this, that Drosophila determination is really, here's what Drosophila has. It has an X and an X, and then a 1. Oh, I'm sorry. That's actually considered 1. 2, 3, 4. 2, 3, 4. So it has, let me not put the homolog there. It has 4 haploid chromosomes. N equals 4, 2N equals 8. And that means you could be XX, 2, 2 prime, 3, 3 prime, 4, 4 prime. Or you could be XY, 2, 2 prime, 3, 3 prime, 4, 4 prime. And remember, the 2 comes from the mom, the 2 prime comes from the dad. The 3 comes from the mom, the 3 prime comes from the dad. The 4 comes from the mom, the 4 prime comes from the dad. All right, so that's what we're talking about here. So the diploid number is 8 and the haploid number is 4. However, chromosome number 4 is really small chromosome. It's really residual, but it's there. But it's really tiny, really residual. It's there. We have to count it, but it has very little information. So sex determination in Drosophila, we won't consider chromosome number 4 when we consider our ratio. What we're really doing is a ratio between the sex chromosome. I'll just do that simpler. It's the ratio between the sex chromosome and the autosomes, and the autosome pairs. All right. So if I'm XX, then my ratio is 1 to 1. That's my ratio because I have 2 X's and 2 autosome pairs. Remember, I'm ignoring, so two X's and two autosome pairs. So that's a 1-to-1 ratio. And if I'm XY, I'm 0.5 to 1, or you could say 1 to 2 ratio. 1 to 2 equals 0.5 to 1. It doesn't matter how you slice that. Because XY would be 1x. There's not another x now, so it's 1 to 2, or 0.5 to 1. So it's really that ratio that helps to determine sex in Drosophila. And what it comes down to is how much protein you're making. All right, so let's take a look at it.
Sex determination in Drosophila is determined by the ratio of X chromosomes to the number of sets of autosomes, X to A. If the ratio is 1, 2 X chromosomes to 2 sets of autosomes, development is set toward femaleness. If the ratio is 0.5, 1 X chromosome to 2 sets of autosomes, development is set toward maleness. The X chromosome contains 3 numerator genes known as the sisterless genes, cis-A, cis-B, and cis-C. An autosome carries the denominator gene called deadpan, DPN. The sisterless numerator genes are expressed to produce protein subunits that can either form homodimers with themselves or heterodimers with the protein subunit produced from the deadpan denominator gene. In females, the expression of the 2 copies of the sisterless numerator genes, one from each X chromosome, results in an excess of sisterless protein subunits compared to the deadpan denominator protein subunits. This results in the formation of many numerator homodimers. The numerator homodimers are transcription factors that activate expression of the sex-lethal SXL gene. In males, there is only 1 copy of each numerator gene, so most expressed sisterless subunits form heterodimers with denominator subunits. As a result, there are not enough numerator homodimers to activate sex-lethal expression. Early in embryogenesis in the female Drosophila, the sisterless homodimers activate transcription of the sex-lethal gene from the early promoter, PE. The pre-mRNA transcribed from the early promoter has 8 exons, but exons 2 and 3 are skipped to produce the mature mRNA consisting of exons E1, 4, 5, 6, 7, and 8. Translation of this mRNA produces the sex-lethal SXL early protein in female embryos. The male embryo does not have enough sisterless homodimers to activate the early promoter of the sex-lethal gene, so no sex-lethal protein is produced in males. Later in embryogenesis, sex-lethal is transcribed constitutively from a more upstream promoter, the late promoter, PL, in all cells, regardless of the X to A ratio. The pre-mRNA transcribed from this promoter consists of 9 exons, L1, E1, 2, 3, 4, 5, 6, 7, and 8. Exon 3 contains a stop codon. In females, the sex-lethal early protein binds to the sex-lethal pre-mRNA and causes regulated splicing in which exons E1 and 3 are skipped. The result is a mature mRNA with exons L1, 2, 4, 5, 6, 7, and 8. Translation of this mRNA produces the sex-lethal late protein. In males, the absence of sex-lethal early protein results in the default splicing of the sex-lethal pre-mRNA. As a result, the mature mRNA includes the stop codon in exon 3. Therefore, no functional sex-lethal late protein is produced in males. The events just described set the switch to either female or male differentiation. A cascade of alternative splicing events follows. In the female embryo, sex-lethal late protein regulates splicing of transformer TRA pre-mRNA. A stop codon containing segment upstream of and contiguous with exon 2 is removed, resulting in a mature mRNA with exons 1, 2, and 3. Translation of this mRNA produces the active transformer TRA protein. In males, default splicing of the transformer pre-mRNA occurs because of the absence of sex-lethal late protein. The stop codon containing exon is not removed, so translation of the mature mRNA does not produce any functional transformer protein. Transformer protein is also an RNA splicing regulator. The target is the pre-mRNA of the double-sex DSX gene. In females, the mature double-sex mRNA encodes the double-sex F, F for female, protein, a transcription factor that represses male-specific gene expression in all cells. As a result, female-specific somatic cell differentiation occurs. In males, the mature double-sex mRNA encodes the double-sex M, M for male, protein, a transcription factor that represses female-specific gene expression in all cells. As a result, male-specific somatic cell differentiation occurs.
Notice that those messenger RNAs are different. Let me show you. 1, 2, 3, 5, 6 is how you become a male.
Transformer protein is also an RNA splicing regulator. The target is the pre-mRNA of the double-sex DSX gene. In females, the mature double-sex mRNA encodes the double-sex F, F for female, protein, a transcription factor that represses male-specific gene expression in all cells. As a result, female-specific somatic cell differentiation occurs.
So this transforming gene codes for the transcription factor that regulates splicing to make this double-sex female gene, where alternative splicing leads to the exons 1, 2, 3, 4, 5, 6. So to become a female, you basically need the double-sex gene exon splicing to be 1, 2, 3, 4, 5, 6. That's the double-sex female protein. You only can get that exon 4 in there if your transforming transcription factor is there. And that transforming transcription factor is only there through this whole cascade of events that we've seen from the beginning. All right. Now remember, the next one, the transforming transcription factor, is not going to be there, and you're going to be 1, 2, 3, 5, 6.
In males, the mature double-sex mRNA encodes the double-sex M, M for male, protein, a transcription factor that represses female-specific gene expression in all cells. As a result, male-specific somatic cell differentiation occurs.
Okay.
Knockout mutants of double sex have both male and female characteristics because there is a lack of repression of both male-specific and female-specific genes.
Okay, no knockout genes. That means it is what it sounds like. We knock out the gene. Now I'm going to talk about how we do that because you don't go in there with a hammer and a chisel, obviously. But there's different chemical ways you can knock out a gene, and it's a huge research tool because how do we know what genes do? Well, let's get it out of there and see what happens to the organism. That's how a lot of researchers have determined what a gene does. So we'll talk about knockout genes later, and we call these knockout mutants, and you see what happens to the organism if you knock out a gene. So definitely coming up in the future. All right, that's sex determination in Drosophila. Sex determination in bees is a haploid diploid story. If you're male, if you're haploid, if you don't have your homologous chromosomes, if you just have 16 single chromosomes, and then you're female if you have your homologous pairs, if you're diploid. So that's how you control sex determination in bees, and that's called the haplodiploid sex determination. As mentioned previously, some environmental factors can affect sex. For example, with this alligator that you see on top here, this alligator up here, if the eggs are incubated at 33 degrees Celsius, uh, you're probably gonna want to know what that is in Fahrenheit. Well, a human body temperature is 37. Room temperature is about 22 to 25. So this is warm, but it's not tremendously warm. 37 degrees Celsius is 98.6, and 22 to 25 is someplace in the 70s, low 70s room temperature. A nice room temperature room like 72 degrees Fahrenheit. So eggs incubated at 33 are males, and eggs incubated above 33 are females, and eggs incubated below 33 are females. The frequency of that is not 100%, 100%. I want to say I don't remember exactly, but I'm going to speculate. If the eggs are incubated above 33 degrees Celsius, then you 100% of the time become a female. If the eggs are incubated a little cool, below 33 degrees Celsius, then you like about 95% of the time become a female. So basically you need 33 degrees Celsius to become a male. These clownfish, these are pretty. The story here is pretty darn cool. Talking about these clownfish now, it's a behavioral thing that determines their sex. First of all, clownfish are protandrous hermaphrodites, which means they can switch from male to female sequentially. All right, that means that at any given time you don't have both sex organs, but you could be a male and switch to a female. And what happens is you have one dominant female living in your sea anemone environment. These guys hide inside the sea anemones. And that dominant female suppresses the development of other females in her group. But she will eventually die. And what happens when she dies is now the suppression of female development in this male stops, and the male organs disintegrate, and the female organs develop, and this male becomes a female. So that's how it works in the clownfish. They're protandrous hermaphrodites or sequential hermaphrodites. They're not male and female at the same time, but they're male and female sequentially. All right, let's talk about some plants. Well, first of all, the plants could be monoecious, which means that you have male and female parts on the same plant. Or the plants could be, and by the way, when you look at so you have a monoecious is like saying hermaphrodite. Monoecious is like saying hermaphrodite. Or you could have dioecious plants, and these are the ones we want to study because we want to study the male plant and see what makes it male, and study the female plant and see what makes it female. So the dioecious a plant is either male, has the male flower, or it's female. And by the way, the male flower, let me remind you, has an anther and a filament, or it's female. And by the way, if you're talking about gymnosperms, because it looks like I have some gymnosperms here, these are angiosperms. Willow is an angiosperm. But if it's a gymnosperm, then you have a male cone, like a pine cone. So it doesn't matter if it's an angiosperm or gymnosperm. So I don't want to get into the botany, so I'm sorry I digressed a little bit. So dioecious means that the plant is either male or female. So if it's male, it has the male cone or the male flower. And if it's female, it has the female cone or the female flower. And you can see that with dioecious plants that the males are like what you're used to in humans. They're heterogametic, and the females are like what you're used to in humans. They're homogametic. But we can't always recognize this in every single gymnosperm or angiosperm that we look at. Sometimes we can't recognize a chromosomal difference in the plants when we're trying to decide what was the genetic determination of sex. So there's a lot of research to be done in plants. Plants do a very funny thing, which makes it hard for us to determine some things. Plants become polyploidy, on purpose, by the way. They have mechanisms to become polyploidy on purpose, so it makes genetic analysis a little daunting. But we're still working on it. All right. What about these homogametic organisms, like the human XX female? So what about that? Well, as it turns out, X is necessary for life. Uh, you can't live without an X. But you really don't want too many X's. So if you're an XX female, then you're going to inactivate one of these X's, and that's called lionization. It's called lionization because Mary Lyon discovered this. Mary Lyon, that was her name. So not just Mary Lyon, but Mary Lyon was a big contributor to this lionization. So X inactivation occurs in female humans, or female mammals, I should say, that where the females are XX. If you're a male and you're XY, you can't inactivate your X. It's incompatible with life. You can't live without an X chromosome. So how do we do this dosage compensation? All right, so there's different mechanisms of dosage compensation. We're going to talk about the human mechanism, the mammal mechanism, where the X chromosome is recognized by a region called the ZIC region. That's a X identifier region. And then there's a gene called the ZIST gene. And the ZIST gene leads to a ZIST messenger, or not messenger RNA, but a ZIST RNA. That ZIST RNA is never translated into a protein, but what happens is the ZIST RNA binds to the DNA and it recruits proteins. So here comes some proteins. It recruits the proteins, and basically what you do is supercoil and condense this X chromosome, and it becomes a bar body. That's basically what happens with this dosage compensation in mammals. You can see a bar body right here. What a bar body is an inactivated X chromosome. Now, how do we discover this? Well, what happens is when you're at about 1,000 cell embryo, so you're a ball of cells, and there's about 1,000 cells in that ball of cells. The cells start inactivating an X chromosome. Now remember, there's 1,000 of these cells. But it's random on what X chromosome they inactivate. So in this cell, they inactivated the maternal X chromosome. In this cell, they inactivated the paternal X chromosome. In this cell, it was the paternal they inactivated. In this cell, it was the maternal cell they activated. Well, what happens is when we have female organisms, we can see a different phenotype because of this lionization. And what we can see is we get things like calico cats. And this calico coloration you can never see in a male. Like you have the white, the yellow, and the black. And you can never see that in the male because the male is either one color or another and never becomes this calico coloration. Well, how could I become this calico coloration in females? Because I inactivated the black in this one. No black. No big B. But I inactivated the yellow in this one. No little b. So so this is how I can become a calico, whereas the male can never be that because they have one X chromosome and that's one gene for coloration. So that's what we get. This coloration, like this tricolor here, you got black, yellow, and white, is called variegation. That's what that's called. So that's what we saw. We wondered why, and that's when we saw that we inactivate the X chromosomes in the embryo at about 1,000 cell stage, and the X inactivation is a bit random. I shouldn't say a bit random; it's completely random. The X chromosome that one cell inactivates is independent and random from the X chromosome that the other cell activates. So uh, I get this variegated coloration in the organism. We also discovered the lionization through some diseases. There's some non-disjunction diseases where chromosomes fail to pull apart. We're going to talk about that at a future date, but I'll just show you this. Join means to put together. Disjoin means don't put together or separate. And non-disjoin means don't separate. And that's where non-disjunction comes from. Don't separate. So if I don't separate my sex chromosomes during meiosis, then what happens is I can get a sperm that has an X and a Y. I failed to separate those guys during meiosis. And that sperm fertilizes an egg that already has an X. So what happens is I get an individual that's XXY. That's called Klinefelter syndrome. That's what that's called. All right, so this individual develops as a male, but they have a bar body. How can a male have a bar body? And by the way, this is the only way males can be variegated as well. Klinefelter males can be variegated because they do inactivate one X chromosome. And Klinefelter males can have bar bodies, but XY males never have bar bodies. We have females. This individual grows as a female. It's still non-disjunction, and I'll talk about non-disjunction at a further time. What happens is this individual grows as a female, but you don't see a bar body. How come you don't see a bar body? Because the individual is X nothing. That's why you don't see a bar body. And you're going to say, well, how could I be X nothing? Well, if this sperm right here got both of the sex chromosomes, remember that you made 4 sperm in that meiotic division. This sperm got none of the sex chromosomes. If this sperm makes it to the egg, I get an individual that's X nothing. So you can see that non-disjunction could occur in either being trisomy or monosomy. So that's a couple words that you don't know yet. The word slipped my mind for a second. That's why the uncomfortable pause. This is called trisomy. This is called monosomy. How come not triploidy? Because not every single chromosome has three copies. You don't have 3 number 1s and 3 number 2s and 3 number 3s. You only have 3 sex chromosomes. The 1 through 22 is normal. But trisomy 21, Down syndrome. Trisomy 13, Patau. Trisomy 18, Edwards syndrome. Turner syndrome, monosomy sex chromosome. So these are ways that we discovered lionization. By the way, when I mentioned trisomy 13, Patau also was involved in the helping to develop the lionization hypothesis. Patau was, but Mary Lyon, she was the major player there. All right, so that's how we discovered the lionization. This is showing you lionization and getting this mouse down here. So you can see the X chromosomes. In this cell, I don't know what I'm going to act. Oh, it's this cell is this cell. So in this cell, I inactivated the big B. But in the cell next to it, that one right there, I inactivated the little b. So I inactivated the whole big B chromosome. And in the other cell, I inactivated the whole little b chromosome. If this was a male, I'm either going to be white or I'm going to be black. Because I only have one X chromosome. I'm either big B or I'm little b. But a female can be variegated, have have black patches on a white mouse, or you could say a white patches on a black mouse, depending on how they turn out, because of this lionization or X inactivation.
X inactivation is a process that mammals use to equalize the gene dosage between males and females. Since normal male mammals have only one X chromosome and females have two, there is a potential imbalance in the expression of genes on the X chromosome between males and females. In female mammals, this imbalance is prevented by inactivating all but one X chromosome in all somatic cells. X inactivation can be divided into three phases: initiation, spreading, and maintenance. Initiation occurs during embryonic development. Each X chromosome has an X inactivation center, or XIC. The number of XICs are counted, and one X chromosome is randomly chosen to remain active while the other is targeted for inactivation. Spreading is the process of compacting and inactivating the X chromosome. A gene designated XIST within the X inactivation center encodes an RNA that coats the X chromosome and promotes compaction, beginning at the XIC and progressing toward both ends until the entire X chromosome is inactivated. When spreading is finished, the compacted chromosome is called a bar body. After the bar body is formed, maintenance begins and continues through adult life. The bar body is replicated just like other chromosomes, but both copies of the replicated bar body remain highly compacted and essentially inactive. Thus, once an embryonic cell undergoes X inactivation, all cells that derive from it will have the same X chromosome inactivated.
Okay, he did say something I want to mention, and that is he said essentially nothing from this bar body is transcribed and translated, and that's true. But there's one exception. I know there could be another exception, but I know that the MIC gene, the MIC gene on even the bar body is expressed, and that MIC gene codes for a protein called CD99. It stands for Cluster of Designation 99, and CD99 is needed in your immune system. It's how the white blood cells get out of your blood and into your tissues to fight off infection. That's called diapedesis or extravasation. So there is a gene on the bar body that is still expressed, and it's expressed on both X's. And so you're going to say to me, so females have more CD99 than males? Yes, we found that females have more CD99 than males. So that's the ZIC gene is how you identify the X chromosomes. The ZIST gene, well, the ZIC is not a gene, it's a region. The X inactivation center is the ZIC region. Within the ZIC region, there's a ZIST gene, and the ZIST gene codes for an RNA that's never translated into a protein. But that RNA complementarily binds to the X chromosome DNA and recruits proteins to help supercoil and condense the X chromosome to a bar body. The bar body is the classic example of heterochromatin. Heterochromatin is supercoiled, highly condensed DNA, whereas euchromatin is loose DNA that allows for gene expression. And you don't have to, the bar body is unique in that the entire chromosome is heterochromatin, and that's kind of unique. But you could have a chromosome with regions of heterochromatin and regions of euchromatin. I mean, chromosome number 1 is like 260 million nucleotide base pairs. So these chromosomes are extremely long DNA molecules. I'm talking about a human, by the way. So you can have a chromosome like chromosome number 1, 260 million base pairs. There could be regions of heterochromatin and regions of euchromatin. But the X chromosome that's inactivated is unique in that the entire chromosome is heterochromatin. All right, let's look at these chromosomes in more detail. Oh, I didn't have to talk about the MIC before. Here it is. The X and Y are homologs, but they don't really line up all that well, meaning homologs. So are they homologous pairs? Yeah, they are. They're kind of homologous pairs. But when you have homologous pairs for like chromosome, all the autosomes all line up very, very nicely. Because they have homologous loci that code for the same characteristic. Remember, they don't have to code for the same trait, just the same characteristic. So in meiosis, the autosomes homologous pairs line up very nicely when they make their tetrad in prophase 1 of meiosis 1. The X and Y line up poorly, but they have some regions of homology. And in these some regions of homology, they do line up. But they do, but they don't line up that well. But what I want to point out is they do have some genes that cross over. Some genes can cross over between the X and Y. It's rare. Crossing over between X and Y is rare. But it can happen. All right? But it can happen. And on the genes that it does happen, they are called pseudo-autosomal genes. The reason why these are called pseudo-autosomal genes is because they act like autosomal genes. They share homologous regions, they line up, they make chiasmata, they cross over. That's just like what the autosomal genes do. Otherwise, the rest of the X and Y crossing over is extremely rare. But there are some pseudo-autosomal genes where crossing over does occur. Okay, just want to tell you about the pseudo-autosomal genes. Most of the diseases we talk about are X-linked. We call them sex-linked, but we do have to talk about X-linked or Y-linked. There are some Y-linked genes we'll talk about, and Y-linked genes are called holandric genes. Amylogenin. Just trying to think how to spell it, because I know some of you guys are going to go out and Google this or Bing it or whatever you do. I'm actually not sure I'm spelling this right. Amylogenin. Amylogenin is a gene on the... Is it this? I'm sure one of you guys is going to grab your phone and Google that. Well, I don't know which spelling is right. That's a gene on the Y chromosome. So it's a holandric gene. We can talk about that. There's a MYC gene on the Y chromosome. That MYC gene codes for CD99, which is involved in your immune system. All right, so holandric genes versus X-linked genes. We'll talk a lot about X-linked genes. Why? Because the X chromosome is huge, has lots of genes. The Y chromosome is pretty darn short and doesn't have very many genes. All right, let's talk about X-linked genes. First of all, X-linked genes was discovered by Thomas Hunt Morgan in the early 1900s. I'm going to spell his name for you. Thomas Hunt Morgan. This was the early 1900s. And because he discovered X-linked genes, he was also the discoverer of more than one type of non-Mendelian inheritance. So what we're going to talk about, Mendelian inheritance, we're going to start talking about things that don't follow the simple dominant recessive pattern. Do not follow the simple dominant recessive pattern. And X-linked traits or sex-linked traits start to violate the mathematics that Gregor Mendel saw. And you'll see that right now. So let's start talking about sex-linked traits.
Various human genetic disorders are said to be X-linked, which means that the defective allele responsible for the condition is carried on the X chromosome. This produces a particular pattern of inheritance because females have 2 copies and males only 1 copy of the X chromosome. X-linked characteristics are therefore expressed more often in males than in females, and as a consequence, X-linked diseases are expressed in more male than female victims. Red-green color blindness is an example of an X-linked recessive disorder because color blindness is not expressed if a functional allele is present. On the Punnett square, we can denote the X chromosome with the normal, functional allele with a black X, and the X chromosome with the defective allele with a red X. The possible progeny are XX, both normal alleles, red X, black X, carriers with one normal allele, black X, Y, normal males, and red X, Y, colorblind males. Note that the genotype must be either red X, female, or red XY to express colorblindness. A female of genotype red X, black X has normal vision and is a carrier for the disorder. Although she will not express the recessive trait, she can pass the defective X allele to her offspring. As this example illustrates, the chromosomal makeup of males makes them more susceptible to X-linked recessive disorders than females. For a daughter to express colorblindness, she must inherit two recessive alleles, genotype red X, but a son who inherits a single recessive allele will have the colorblind genotype red XY. As a consequence, colorblindness is much more common in males than in females.
All right, I'm going to play that again.
Various human genetic disorders are said to be X-linked, which means that the defective allele responsible for the condition is carried on the X chromosome. This produces a particular pattern of inheritance because females have two copies and males only one copy of the X chromosome. X-linked characteristics are therefore expressed more often in males than in females, and as a consequence, X-linked diseases are expressed in more male than female victims. Red-green color blindness is an example of an X-linked recessive disorder because color blindness is not expressed if a functional allele is present. On the Punnett square, we can denote the X chromosome with the normal, functional allele with a black X, and the X chromosome with the defective allele with a red X. The possible progeny are XX, both normal alleles, red X, black X, carriers with one normal allele, black X, Y, normal males, and red X, Y, colorblind males. Note that the genotype must be either red X, female, or red XY to express colorblindness. A female of genotype red X, black X has normal vision and is a carrier for the disorder. Although she will not express the recessive trait, she can pass the defective X allele to her offspring. As this example illustrates, the chromosomal makeup of males makes them more susceptible to X-linked recessive disorders than females. For a daughter to express colorblindness, she must inherit two recessive alleles, genotype red X, but a son who inherits a single recessive allele will have the colorblind genotype red XY. As a consequence, colorblindness is much more common in males than in females.
And matter of fact, color blindness is only possible in females if the dad is colorblind. So before we go on, I know that video just showed you the using the red X being the recessive allele. But a lot of times what we do is we'll say, all right, you got a big C and you got a little c, and that's a carrier female. Or you have an X little c Y, that's a colorblind male. But Thomas Hunt Morgan, what he did was he didn't use big little letters. He used plus being the wild type and the absence of a plus being the mutant or less frequent type. So what do I mean by wild type before we go on? Well, what I mean by wild type is the type most commonly seen in nature. All right. So wild type is just what you usually see. Wild type. For example, with fruit flies, red eyes is the wild type. Red eyes is the wild type. All right. And it turns out that the red eyes is on the X chromosome. We're going to talk about that right now. And Thomas Hunt Morgan didn't say this. He didn't say X big R, X little r. What he did was he said XW plus XW. That's a heterozygous red-eyed female. Well, how would you be a white-eyed female? To be a white-eyed female, you'd be XW, XW. How would you be a white-eyed male? XWY. How would you be a red-eyed male? XY. W plus Y. So he changed the nomenclature a little bit. It's not hard after you're, well, I shouldn't say that. It's a little disconcerting at first because you're probably already used to using, big R, big T, little t. And now you're changing to this plus, lack of plus uh, uh, mechanism. And it's just what you have to do. All right. All right, so remember, W plus is red, and it's the wild type. And to be white-eyed, you have to be WXWY or XWXW. All right, so what did he do? This is what he did. He bred a true breeding white-eyed male with a true breeding red-eyed female. Now, what do I mean by true breeding? That means homozygous. Well, is this really homozygous? Well, it's hemizygous. We call it hemizygous because this guy only has one eye color gene because it's on the X chromosome, and he's a male, and he only has one X chromosome. So we call it hemizygous. All right, what did he get? Well, he got all heterozygotes or hemizygotes, in other words. So he got this, a red-eyed male. He got this, a red-eyed female. He got this, a red-eyed male. And, whoop, no, that's actually who he's breeding. This is all he got, was a red-eyed male and a red-eyed female. That's what he got. If you wanted to do the Punnett square, and I encourage you to, I'm not always going to do this for you. Let me say that again. I'm not always going to do these Punnett squares for you, but I'll do this one. So go ahead and do it. You get XW plus XW. That's a red-eyed female. She's heterozygous, but that's a red-eyed female. XW plus Y. That's a red-eyed male. XW plus XW plus, or XW plus XW, I'm sorry. Another heterozygous red-eyed female. And another red-eyed male. So we got 1-to-1 red-eyed females with red-eyed males. 1-to-1. Okay, then what he did was this is the F1 generation. Then what he did was he mated the F1 generation together. Sound familiar? Yeah, you've already done this in the Mendelian genetics. So he mated this F1 generation, and this is what he got. You're going to say 3-to-1 ratio is what I expect. I agree. I agree that 3-to-1 ratio is what you expect. But it should be a 3-to-1 ratio, and you should have equal numbers of white-eyed males and white-eyed females. Sex should not matter whether your eyes are white or red, unless the gene is on the sex chromosome. And what he found was only the males were white-eyed. Only. He never got a white-eyed female, ever. And by the way, turned out he really didn't get a 3-to-1 ratio. He got a 4-to-1 ratio. And that's because white-eyed individuals don't survive as long. They have less vigor. They don't last as long. It's not a lethal gene. Being white-eyed is not lethal, but it has less survivability. So the ratio actually changes because of that. So he actually discovered a couple things here. Because of this, because he saw that all his white-eyed progeny were all male, and that's weird. He determined that it was on the X chromosome. He also discovered that being white-eyed you had less survivability. All right, then he mated his F2 generation together, and he mated his white-eyed male with his heterozygote female. All right. Oh, actually, how do you know that's heterozygote? Because he mated the F2 with the F1. Because if that was F2 by F2, you wouldn't know if that female was heterozygote or homozygote. Here's a homozygote red-eyed female from the F2 right there. So he didn't mate the F2s together. What he did was mated a white-eyed F2 male, and all the males were white-eyed. All right. Well, actually, that's not true. Not all of them. Here's a male that's not. Here's a male that's red-eyed. But he did mate a white-eyed F2 male with the F1 generation female, and he knew all of the F1 generation were heterozygotes because the parental generation were true breeders. And this is what he got. That's what he got. This is further evidence for the X-linked nature of the eye color gene. Why? When I say why, I mean I'm asking the question, why? Because the only way to get a white-eyed female is if the dad is white-eyed. And he did get a white-eyed female. All right, so that was Thomas Hunt Morgan's experiment. This is just saying to you, I left off the X's. No, I actually didn't, not on purpose anyway. Excuse me. If it wasn't X-linked, here's your white-eyed, but you would expect equal numbers of males and females to be white-eyed. So it's 3 to 1. If you had 1,000 offspring, you would have 250 white eyes, and you would expect just about half and half to be, 125 of them to be male and 125 of them to be female. That's what you would expect. But that's not what he saw. He saw that his white eyes were all male. All right, so then he does a test cross. Remember what a test cross is. This is really just a test cross. He takes a white-eyed male, took it from the F2 generation, and he mates it against a heterozygote F1 female, red-eyed female from the F1 generation. He knew she was heterozygote, and he gets white-eyed. All right, he gets white-eyed. I forget what I'm trying to tell you here. Oh, I'm not trying to tell you anything. This is what he did, a test cross, and he knows that this should actually be. Well, it actually should be half and half, and it is. It's a 1 to 1 to 1 ratio. It's a 1 to 1 to 1 to 1 ratio. Oh, I know. But what happens is, if this is X-linked, which is what he thought up here, if this is X-linked, then the only way to get a white-eyed female is by mating with a white-eyed male with a heterozygote female. That's the only way to get a white-eyed female. And one of these is a female. When I say one of these, and it's just a percentage, one of those is female, and one of those is male. All right. By the way, we're going to do a reciprocal cross in a second, because the reciprocal cross will absolutely put the nail in the coffin about this X linkage. All right, so this talks about, well, he didn't really get a 3 to 1 ratio. How come we didn't get a 3 to 1 ratio? We got about a 4 to 1 ratio. Well, we already said it, and that's because the white-eyed fruit flies have reduced survivability. So this is less than 1. I should say it's less than 1/4. Because this is 25%, this is 25%, this is 25%. So you actually have 50% heterozygotes, 50% heterozygotes right here. No, you don't, not really, because this is a hemizygote, not a heterozygote. This is hemizygote, and this is 25%. Well, as it turns out, this is only about 15% because they don't survive as well. So when he looked at his ratio, he wasn't getting 25% of these white-eyed males surviving. He was getting less than that. So it changed my ratio from 3 to 1 to about 4.5 to 1. 4 to 1, 4.5 to 1. Reduced survivability, that's all. And by the way, when he did this cross, he didn't get 1 to 1 to 1 to 1. He got about 1.5 to 1.5 to 1 to 1. That's what he got. And why? Because these guys have reduced survivability. These white-eyed individuals, white eye, have reduced survivability. So he got close to 1 to 1 to 1 to 1, but not quite. And you can see that here. This is not 1 to 1 to 1 to 1. It's about 1.5 to 1.5 to 1 to 1. You guys can do the math if you want. All right. To absolutely determine this X linkage, he did a reciprocal cross. Remember what he did before. What he did before is he took a white-eyed male from the F2 generation, and he crossed it with, well, this is actually from the parental generation. Well, it doesn't really matter to show you the reciprocal cross. And if you take a white-eyed male from the F2 generation and you cross it with a fruit fly from the parental generation that is a true breeding red-eyed female, here's what you're going to get. You're going to get all red eyes. All red eyes. Even your males have red eyes. Well, do the reciprocal cross to that. And instead of having a white-eyed male, have a white-eyed female. This is a white-eyed female. Remember, she had to come from the F2 generation. And then take a red-eyed male. That's just the reciprocal of what you did up here. This was a white-eyed male and a red-eyed female. Down here I have a red male and a white female. And look what you get. What I get is a 100% white-eyed males. And that's a reciprocal cross. The only way to get that is if this is sex-linked. That's the only way to get this. If you set this up and do the Punnett square and take out the X and Y's and do just the type of autosomal gene, you won't get 100% white-eyed males. It'll be a mix of males and females doing this reciprocal cross. So this was determinative. He absolutely was convinced now that this eye color gene was on the X chromosome.
The chromosome theory of inheritance states that genes are located on chromosomes. In 1910, Thomas Hunt Morgan crossed certain strains of the fruit fly, Drosophila melanogaster, and discovered sex linkage, the presence of genes on the sex chromosome of eukaryotes. Morgan studied a gene affecting eye color in Drosophila melanogaster. The wild-type eye color is red, while the mutant eye color is white. This gene is located on the X chromosome. Females may have 2 wild-type alleles, red eyes, 2 mutant alleles, white eyes, or be heterozygous, red eyes. Males have only 1 X chromosome, so they may have either 1 wild-type allele, red eyes, or 1 mutant allele, white eyes. There is no equivalent gene on the Y chromosome in males. Morgan crossed a red-eyed female with a white-eyed male. The female is homozygous for the wild-type allele, with the genotype W+W+. The male is hemizygous with the genotype WY. The female produces only 1 type of gamete, with an X chromosome carrying the W+ allele. The male produces 2 types of gametes, one with the X chromosome carrying the W allele and the other with the Y chromosome. The male and female gametes fuse, leading to the development of the F1 generation. All of the F1 females are heterozygous and have red eyes because the W+ allele is dominant to the W allele. All of the F1 males receive the X chromosome with the W+ allele from their mother and have red eyes. Genotypically, they are W plus Y. Interbreeding the F1 flies produces the F2 generation. The F1 female produces 2 types of gametes, one with the X chromosome having the W+ allele and the other with an X chromosome having the W allele. The F1 male produces two types of gametes, one with an X chromosome having the W plus allele and the other with the Y chromosome. Fusion of the male and female gametes leads to development of the F2 generation. There is a 3-to-1 ratio of red-eyed to white-eyed flies.
Yeah, you expect that, remember, but here's what you don't expect. You do not expect all of your white-eyed flies to be male. You expect equal numbers of males and females. So it's not the 3-to-1 that throws you off, that threw Thomas Hunt Morgan off. It was the fact that his 25% of his flies that had white eyes were 100% male. He had no white-eyed females. That threw him off.
However, if you look at the sexes separately, you see there are differences based on sex. The females are all red-eyed, one half with the genotype W+, and one half with the genotype W+W. The males all receive the Y chromosome from their fathers, so their phenotype depends upon the X chromosome received from their mothers. Of the F2 males, one half are red-eyed with the genotype W plus Y, and one half are white-eyed with the genotype WY. Morgan also crossed a white-eyed female with a red-eyed male. This is the reciprocal of his first cross. All females produce one type of gamete having the W allele on their X chromosome. Males produce two types of gametes, one with the W plus allele on the X chromosome and the other with the Y chromosome. Fusion of these gametes produces red-eyed females, all having the genotype W plus W, and white-eyed males having the genotype WY. This result is distinctly different from Morgan's earlier cross. Interbreeding of the F1 flies produces the F2 generation. The W plus W red-eyed F1 female produces two types of gametes, one with an X chromosome with the W plus allele, and the other with an X chromosome bearing the W allele. The WY white-eyed F1 male produces two types of gametes, one with the X chromosome bearing the W allele, and the other with the Y chromosome. Fusion of the F1 gametes leads to development of the F2 generation. The overall ratio is one half red-eyed and one half white-eyed. However, if we look closely, we see that one half of the F2 females are red-eyed and one half are white-eyed. The males all receive a Y chromosome from their fathers, so their eye color depends on the X chromosome inherited from their mothers. One half of the F2 males are red-eyed and one half are white-eyed. Comparison of these reciprocal crosses demonstrates that for an X-linked recessive trait, there will be apparent phenotypic differences between males and females in both the F1 and F2 generations. Morgan's discovery of X-linked genes strongly supported the chromosome theory of inheritance.
And why would that be? Because if the eye color gene is on a sex chromosome, then genes are on chromosomes. I want you to remember something from a previous lecture. The Boveri and Sutton experiments and Weissman, Nigeli was a contemporary of Gregor Mendel. I mean, he lived at the same time as Gregor Mendel, and Nigeli actually tried to get Gregor Mendel to abandon pea plants and study some other things. But what happens was Mendel's work went largely forgotten for 40 years. You know, he did his work in the 1850s. So for about 40 years, 50 years, his work was largely forgotten. But then it was rediscovered in the early 1900s, and there were some big players, Correns and Boveri and Sutton, and you don't have to memorize these names. But when his work was rediscovered, Thomas Hunt Morgan in the early 1900s was one of them that was doing genetic work, And this does support chromosome theory of inheritance because if the eye color gene is on the X chromosome, well, the X chromosome's a chromosome. Genes are on chromosomes. However, we still didn't know if the gene was a DNA or a protein. We knew chromosomes were made of DNA and proteins, but we didn't know what the genetic material was. And we really don't know that until 1944. So we have a suspicion in 1928, but we really don't know for sure that DNA is the genetic material till 1944. But now we know the genes, unequivocally, we know the genes are on chromosomes, the chromosomal theory of inheritance. All right, in humans, we use pedigrees to study our genetics, and this is an X-linked pedigree. So you've already seen one pedigree, but this is an X-linked pedigree. Let me just remind you that boxes are males. That's a male. Circles are females. These are the kids, and this is the marriage. So this is the paternal generation or the first generation. This is the F1 or second generation. This is the third generation or F2. This is the fourth generation or F3. You can see that. You can see a marriage right here. The daughter of the parental generation married this guy. When you're half colored in, you're a carrier. So if I'm talking about Duchenne's muscular dystrophy, which that's what this is. This is on the X chromosome. To have Duchenne's muscular dystrophy, you got to be little d, little d. So you could be normal and be a carrier, or you could be normal and not be a carrier. If you're half colored in, you're big D, little d. If you're completely colored in, that means you're diseased. You're little d, little d. On a test, you're going to have pedigrees to solve. What I suggest you do in the beginning is you simply write down what you know. All right? That's what you have to write down. And by the way, I probably shouldn't write it like this because these aren't autosomes. So let me just write it like this. X little d y, X big D, X little d, X big D, X little d, X big D, X little d, and so on and so forth. All right. This is X big DY. So write down what you know, and then you can start solving for what you don't know. You certainly can do that. All right. So this is a pedigree of it. But I want you to look at the pattern of the pedigree. Do you see any diseased females? Not carriers, but diseased. And the answer is no. You don't see a diseased female. There is a couple cases where you might have seen a diseased female. If this guy right here married a carrier, then they could have had a diseased female. They could have. Because the female is going to get an X from the dad, which you know that X is little d, because that dad's X little dy. And the mom over here that guy married is X big D, X little d. 50% of the time, that other X little d is going to come over. That could have happened. But it didn't happen in this pedigree. So with X-linked diseases, you usually see a pattern of males being diseased. That's what you usually see, and not females. And I should say X-linked recessive patterns. This is X-linked as well. It doesn't look like they're going to draw the X for you. They're just going to say little c, little c. Remember that they probably should say X little c for a colorblind female. X big C, X little c for a carrier female. And X big CY for a normal male, and X little CY for a colorblind male. But it doesn't look like they are. They're just using little c. But you know that female or male from the circles and the squares. Note the condition only occurs in men. That's all they're telling you. All right. I hope that's apparent to you. This is Duchenne's muscular dystrophy and color blindness. You could do a dihybrid cross now. I mean, I don't know if you want to I guess you could have a pedigree where you're looking at Duchenne's muscular dystrophy and color blindness, but this is just showing you it's wrapping up both of them for you. This is showing you the reciprocal cross. This is a dog with Duchenne's muscular dystrophy. It's X-linked in dogs as well. So they do a reciprocal cross. So what did they do? They took a male who had Duchenne's muscular dystrophy and mated it with a female that was normal, not a carrier at all, but normal. But now you have carrier status right here. Now you have some females that are carriers in the F1 generation. Here you have a normal male, and here you have a normal male. All right, so that's the first cross. And then you do the reciprocal cross. Now you have a female that has Duchenne's muscular dystrophy, and you have a normal male. And look at, you get a male with muscular dystrophy. You get a male with muscular dystrophy. Okay, so that's the reciprocal cross. We've already gone through that. All right, folks, that's it. See you in Chapter 5.